Hello, everyone, and welcome back. We'll now move on to our next session, High-Performance Antibody Discovery to Power Biomarker Studies, and I'm pleased to welcome April Livengood. April is Director of Strategic Business Development and Collaborations at Aviva Systems Biology. She has a PhD in Molecular, Cellular, and Development Biology from the University of Colorado at Boulder, and has conducted translational cancer research as a postdoctoral fellow at the University of California, San Diego. She has an extensive background in translational research, genomic medicine, assay and product development, and leadership of interdisciplinary teams. Prior to joining Aviva Systems Biology, she's led many internal and external initiatives at San Diego area biotechs, including EMD Millipore, Pathway Genomics, Illumina, and Thermo Fisher Scientific. A very warm welcome to you, April, and please take it away.
I appreciate the opportunity to speak today along with this great lineup of speakers. I'm presenting on behalf of Aviva Systems Biology, where I'll talk about our high-performance antibody discovery to power biomarker studies. So at Aviva, our goal is to develop recombinant antibodies that can help enable and power biomarker studies. We have a focus on developing highly sensitive and specific antibodies, which are a key tool to identify and study protein biomarkers. And we're very committed to developing high-performing antibodies for challenging targets. So what do I mean by challenging targets? There's a lot of reasons why it might be challenging to develop antibodies and assays for a protein biomarker. It could be, for example, some proteins are detected at very low levels in biofluids such as plasma, serum, or CSF. So you need a very high affinity antibody and assay to detect those. Also with, in some cases, you need a really broad dynamic range as well. There can also be challenges like if it's only found in a very rare cell type. Also, there can be challenges around finding the right specificity. So if you have a protein isoform that's very similar to other proteins, then finding something that specifically detects the isoform of interest and doesn't cross-react with other proteins is important, or maybe distinguishing a form of the protein with a single post-translational modification or a single mutant amino acid. So not only the protein target itself, but also the type of cells, fluids, or samples you're working in means you can encounter a variety of challenges when you're trying to make the right tool. And until you have the right tool, you can't really ask the questions you want to better understand the role of a biomarker in disease progression, monitoring therapeutics, and other indications. So we've developed a proprietary antibody development workflow at Aviva that helps with the rapid identification of antibodies for challenging targets. And today I'll walk you through that as well as give you an example of where we used it to make a high affinity antibody pair to detect the biomarker progranulin in clinical samples. First, let me give you a brief introduction on Aviva. So Aviva is a Southern California company based in San Diego. We've been around for over 20 years. Very early on, the company launched tens of thousands of polyclonal antibodies to cover a large percentage of the proteome, basically providing antibody tools to help study a lot of different proteins, including rare ones where there may not be many tools available. The company expanded its product offering to include a lot of ELISA assays, recombinant proteins, and other reagents to support researchers. And for this past several years, there's been a real focus on developing its recombinant antibody pipeline and capabilities to make high-performing antibodies and assays with those antibodies. More recently, we've also shifted our focus more to antibodies and assays to serve translational research, biomarker studies, and diagnostic applications. I should also mention we acquired Genway Biotech almost a decade ago now, and that brought in our GMP manufacturing capabilities. We have a lot of custom services around antibody, protein, and ELISA development, and we are also ISO certified. So you may be familiar with recombinant antibodies, but I want to give you a little bit of a background on the importance of these reagents and why there's really been a shift toward trying to use those, especially for key antibody tools and assays. Maybe you've used polyclonals, maybe some hybridoma-based monoclonals, or even recombinants in your research. But if you're really relying heavily on your antibody as a tool, you need it for important studies. Maybe you're developing an assay with it, and you want the performance at day 0 to be the same 5 years later, and also not worry about the stability of supply, then really recombinant antibodies are the way to go. They have the highest lot-to-lot reproducibility, easily scalable production, stable long-term supply. If it comes to something like a monoclonal that's produced from a hybridoma cell line, you have to worry about contamination and genetic drift and other things that can influence the performance or even result in the loss of your antibody in worst case scenario. With the recombinant, you don't have to worry as much because in addition to physical storage, you also have, in a sense, digital storage because you have the sequence of the antibody. So we see that a lot of assay developers that we work with, and just in the field in general, there's been a real shift to recombinant antibodies, especially for key antibodies and assays that rely on those antibodies to shore up your supply and your performance. So now I'll talk about our recombinant antibody development workflow. I'll give sort of a high-level overview of the workflow, and then some of the key steps I'll describe more in detail later. This workflow also can be modified depending on the type of antibody target we're going after and the type of application and sample type that we need those antibodies to work in. So first, I'll walk you through an example of how we would develop antibodies to ultimately find antibody pairs that can work in a sandwich type immunoassay, such as an ELISA. There's other sandwich immunoassay platforms as well, but that's kind of a very common one you may be familiar with. So we start with AI-informed antigen design. Often we have protein fragment immunogens, and we immunize rabbits. We find that developing antibodies in rabbits yields high affinity specific antibodies. There's a very diverse antibody response in rabbits, so that's our preferred system for generating the antibodies. So we let Mother Nature do its work, and then a few months later, we want to isolate the cells producing antibodies specific to the protein target, or technically the immunogen that it was generated against. So we do single B cell sorting at this stage to isolate antigen-positive B cells. And from those cells, we do small-scale antibody clone production. We can make small amounts of antibody from 100 or even hundreds of different cells. So we have a lot of antibodies at this stage, and we can do high-throughput screening on the Carterra LSA. This is a high-throughput surface plasmon resonance instrument. So we can do kinetic analysis looking at affinity. If we're developing antibody pairs, we may also do an additional experiment on there known as epitope binding to help identify antibodies that will pair well together. And then based on the results from our early test with hundreds of antibodies, we pick the best candidates to move forward to do our sequencing, look at diversity analysis, and then pick the best candidate antibodies to then move forward to our functional testing. So we do scaled up expression, and we can develop several or 10, 15, 20 antibodies that we then go on to do our functional characterization. So in this case, if we're trying to develop an antibody pair, we would do a lot of pairwise testing to find the best antibody pair, making sure the performance works well with good dynamic range and on the samples of interest. And ultimately, we can find the best pair to progress to full immunoassay development. Now, for example, if we're trying to develop an antibody for a different application, perhaps to work in cells or tissue, a lot of the workflow is the same, but we would modify it some to increase get better results and better customize it to the type of project. So, for example, if we want to have an antibody that works on cells by immunocytochemistry, after we do the initial steps, in addition to the kinetic analysis, we would also do small-scale application testing for the application or applications that we're most interested in. Again, then we would pick the best candidate antibodies based on that data and performance to do our sequencing and diversity analysis, and pick the top antibodies to make more antibodies to scale up the expression and do a lot more characterization and final application testing. So, for example, making sure it works well in the right sample types, have positive and negative controls for specificity, make sure it has the right sensitivity, and do all of that on a, going from hundreds of antibodies to perhaps several, where we can then do the deeper analysis and find the best performing antibody or antibodies. So as you can see, we can modify a little bit based on the type of application. I do want to give a little bit more detail on a few of the steps of our workflow. So we have structure-guided protein antigen design. So the team has really developed a lot of expertise around designing the immunogen or antigen for these projects. It's influenced by expertise and data from a lot of past antibody development projects. We also factor in the AlphaFold predicted protein structures, a lot of different biophysical metrics, and also antigenicity predictions. So we both want more of a protein that has a similar structure to native protein that's also going to be antigenic in the rabbits. And of course, we factor in other important biology that influences where an antibody should or shouldn't bind on that target protein molecule. So we really want to get this right at the beginning, because if we want to get good antibodies at the end, we really have to make sure our antigen design sets us up for success. So then we would immunize rabbits with our immunogen, and then after that, as I had mentioned, if we want to isolate the small percent of cells from either spleen or PBMCs that express our antibody of interest, we have a several parameter FACS panel, including double staining for antigen positivity. So this helps us get the small subset of cells that we want from that large cell population, and also reduces the false positives. And we can identify 100 or hundreds of antigen positive B cells in a day. This is really important, especially if we think it's going to be really challenging to find an antibody with the properties we want. Like we need a really high affinity, or it's really challenging to get the specificity we want. Then having a lot of different antibodies to screen and down select from increases our likelihood of success. So if we have the cells, then we need to make antibody protein from those cells. And if there's hundreds of cells, then obviously the scale of this could get pretty out of hand pretty fast. So the team developed an optimized high throughput, low volume antibody expression approach. It can be done in a 96-well format, and it utilizes single cell PCR and plasmid-free protein expression. And this is done in mammalian cultures. And so by isolating a lot of cells and then being able to make antibody from hundreds of cells, we really, again, just want to reinforce that we have a lot of different antibodies to start with, and this increases the likelihood of that in the end we get ones that meet our performance criteria. So now that we have that small amount of antibody, hundreds of different antibodies, fortunately, we were able to leverage the Carterra LSA which is a surface plasmon resonance instrument. And we can do high throughput kinetic analysis on this. So it has fast turnaround time, and it only requires a low volume of sample, which is what we would have. So that works out well. It can be 96 or 384 antibodies at the same time. And typically the antibodies are immobilized, and then you come back with a protein analyte of interest, and you can get your on and off rate and your dissociation constant, so you can understand the affinity of the antibodies. But not just for your protein target of interest, but if you're looking for particular specificity, you can not only flow on your protein of interest and look at the on and off rate, but you can come back with related proteins, for example, negative controls, or do a counter selection if you want your antibodies that bind a particular isoform but not a related one, or a particular post-translational modification, but not something without it, or plus or minus a point mutation that results in a single amino acid change. So we really get a lot of information on hundreds of antibodies around affinity and some initial specificity data as well. And then if we are developing an antibody pair, and let's say we have a large protein that we want to have antibodies that can bind cooperatively, we can do epitope binding, which is basically you immobilize all the antibodies, then you bind your protein molecule, your potential biomarker, the protein you're interested in, to the antibodies, and then come back with each antibody one at a time. And at a high level, what's happening is you're looking for antibodies that can both bind your protein at the same time. So they could potentially pair together, and they would be in different bins. But if two antibodies compete for binding, they can't bind the protein at the same time, then they would be in the same bin. So with all of these antibodies and these analysis we do on the Carterra, we can look at affinity, we can have some initial specificity testing, and if needed, we can also do epitope binding and then pick the best antibodies from the hundreds to then move forward to do sequencing, scaled up expression, and then functional testing. So that kind of gives you a flavor for our workflow and how it can be fine-tuned for different kinds of projects. Now I'd like to show you an example from one of our projects. So we developed antibodies and ultimately antibody pair and an immunoassay for the biomarker progranulin. Progranulin is implicated in several diseases, including Alzheimer's disease, Parkinson's disease, and ALS. Mutations in progranulin, heterozygous mutations, actually have been shown to cause a highly penetrant form of frontotemporal dementia, and it's also studied as a potential biomarker. Because of the role in the monogenic form of frontotemporal dementia, there are a lot of biotech and biopharma companies that are already trying to develop progranulin therapeutics to restore the levels of wild-type progranulin, either directly or indirectly, and several of those are shown here on the right. And so using the workflow that I described, we wanted to develop progranulin antibodies, so we designed our antigen using the approach I described. We immunized rabbits, and then after that, here's the results from some of the different workflow stages. So we did the single B cell sorting, and we ended up with over 200 antigen positive B cells. Then using single cell PCR and plasmid-free expression, we were able to amplify small amounts of antibody, about 150 different potential antibodies. We did our Carterra analysis with these, and over 100 of those bound not only our protein fragment immunogen, but also full-length mammalian progranulin protein, and we were also able to do epitope binding. So in the next two slides, I'll just show you some representative data from this project. So again, as I mentioned, we isolate B cells, then we make small amounts of antibody, and then we can analyze hundreds of them on the Carterra instrument. So the data here is not from all the antibodies, but just from a subset to give you an idea of the type of results we get. So in A on the left, the spot diameter is actually the relative expression level of the antibodies. Then in B, the antibodies that actually had reasonable affinity for both our immunogen protein fragment and the full-length mammalian expressed protein are still blue. And then if we look at the affinity for the mammalian expressed full-length protein, we can see some of these are really strong binders, such as in very dark blue, or weaker binders, such as in red. And then we had also done the epitope binding, and this here is a dendrogram, which is another way to depict that sort of data. And antibodies are grouped into different clades. And if they're in the same clade, they probably have overlapping binding sites and they would not likely pair together. If they're in different clades, they have a much more likelihood to work well as an antibody pair. So from analyzing 100 plus antibodies on the Carterra, we looked at specificity, we looked at affinity, and then also could pick antibodies from different clades to down select to a manageable number of antibodies to sequence, and look at the sequence and diversity, and then do scaled up expression, because we have a lot of data suggesting what are good antibodies for potentially working as an immunoassay pair in an ELISA, but we still need to do the functional testing with the right sample types and whatnot to find the best pairs. So one more slide of data shown here. This is just some representative data. So we tested a lot of different antibodies, about 12, and then these are some of the pairs that came out of that testing. And clearly some perform better with more dynamic range than others. We also checked performance, including in some patient sample types, to narrow down to our best antibody pair. And for that, we then developed it into a chemiluminescent ELISA. And, as you can see, we also looked at performance in some different patient-relevant sample types. And also, we tested in both control and neurodegenerative disease patient samples, both serum and CSF, to get some preliminary data around how it works in patient-relevant samples. So at this point, this is just a little bit of the data and the work done to develop this ELISA, but we have a chemiluminescent progranulin ELISA. The next step is we want to assess the clinical utility in collaboration with clinical researchers, and we have some things lined up. We're particularly excited about testing in frontotemporal dementia patients, both mutation positive and negative, but there's certainly a lot of other disease indications where our progranulin assay is a useful tool, and we'd like to see its performance in those other disease settings as well. So taking a step back, I hope I was able to show you how our recombinant antibody workflow can help generate tools that can be used in the study of challenging protein biomarkers. If you'd like more details about our workflow or our progranulin project, you can certainly use this QR code on the right and download our case study. And I'd also like to highlight we do have more emerging and established biomarkers in our antibody and assay development pipeline. So I appreciate having the opportunity to speak with you today about our workflow, our pipeline, and some of the work we've been doing. If you need any help developing antibody or assay tools, please feel free to reach out. There's an email here. And then also, if you're interested in collaborating, here is my contact information as well. Feel free to reach out. And again, I very much appreciate the opportunity to meet with you today and speak about our work, and look forward to hearing the rest of the talks.